Welcome, everybody. I know the program indicates that Betsy Humphreys will be up here, and I promise you that Betsy will be in a minute. I'm Glenn Campbell, the chair of the Friends of the National Library of Medicine, and I wanted to take this opportunity to welcome Jonna Mazet, our distinguished speaker today. You will hear more about in the intro from Betsy about Dr. Mazet, but I wanted to welcome Dr. Lindberg and Mrs. Lindberg, our special guests here today, and also just comment that the Friends are very pleased to host a reception afterwards that we hope you'll all join us after the lecture. We can't imagine that the National Library of Medicine or the Medical Library Association would have any rivals in providing a better opportunity for providing a lecture on biomedical communication. These institutions have been at the forefront, the association and the library are at the forefront of improving biomedical communication throughout the world for all participants, and it's a pleasure to be here today. Last night, we heard importance of health literacy with regard to genetic literacy in another lecture, and I look forward to this one today. So again, thank you all for being here, a hearty welcome, and now the Acting Director of the National Library of Medicine, Betsy Humphreys.
So let me add my welcome to Glenn's, and to say that it is a great pleasure to have all of you here, and I think we're being joined by many people via the video cast for what is the 33rd Joseph Leiter NLM MLA Lecture. The lecture was established in order to stimulate intellectual liaison and discussion between the NLM and the Medical Library Association for topics related to biomedical communications, always interpreted very broadly. Joe Leiter had a long and distinguished careers at the National Cancer Institute, and then a second one here at the National Library of Medicine, where he led the development of programs that had a profound positive impact on medical librarians and on the delivery of biomedical information worldwide. As you might guess from this picture, Joe loved vigorous discussion, not to mention debates or even arguments, which is what the other people in the discussion sometimes felt they were engaging in. Among many other things, he was a great mentor to me personally and a great supporter of the advancement of both women and minorities in the professions. So I know that he would be delighted, were he here, to see that we have such a distinguished lecturer, Dr. Jonna Mazet, who I will not read all the degrees, but she obviously is both a veterinarian and has a PhD, and she is a professor of epidemiology and disease biology and the executive director of the One Health Institute at the University of California, Davis. Our outgoing chair of the Board of Regents is from Davis, and she informed me that you are definitely one of their superstars. She has really now been engaged in the management of really huge and very interesting international programs that are looking at the relationship of disease transmission among wildlife, domestic animals, and people, and the ecological drivers of disease emergence. This is, of course, a topic of great interest to all of us, and the work in this area is also a great source of data and information that is pouring into the National Library of Medicine and into GenBank and many of our resources every day. I think that we all have a great deal to learn from Dr. Mazet, and we're really very grateful to her for coming to speak to us.
Well, thank you for having me. It's such an honor to be here today. Of course, I'm a beneficiary of medical library libraries and been able to build my career. I wouldn't be able to have learned and studied and continued lifelong learning without them, and as well as the NIH for our own health, but also for our studies and being able to move forward the needle for emerging infectious diseases. So today I want to talk a little bit about moving from this reactive place where I'm sure you're all feeling the burn of the reactive place of responding to things like Ebola and Zika and sort of living in this age of fear, I guess. And our goal with this project, and I think as a global health community, is to have the information deposited in the right places, curated in the right ways, to be able to not be afraid, to understand what the risks are, and to move forward in an informed manner. So a few years ago, going on 7 years ago, a team of scientists decided to respond to a challenge from the U.S. Agency for International Development. And that challenge was to try to stop, before they got started, zoonotic diseases, or those diseases that spill over from animals to people. And most of the emerging infectious diseases that we see today, like Ebola, MERS, all of those others, even Zika, originated in an animal host. And so what we're trying to do is sort of figure out how to stop them from spilling over, or if that's not feasible, stop them from expanding their range if they do have local spillover events. Now, you might imagine that if we are successful with that, no one will know. So what happens to NIH funding if no one knows you are successful? You don't get any more. I guess that would be a great problem to have, right? The world would be in a much better situation if we were able to stop these. But it's a pretty crazy call. It's a pretty crazy challenge to be able to stop everything before it starts. Yet we endeavor to try. So I'll tell you a little bit about how we started to chip away at this issue, and what the future may look like if we collectively as a health community could move forward in a more progressive manner. So if you take a look at this graph, we're looking at an epidemic curve. So the blue epidemic curve is for domestic animals getting sick from a pathogen that might spill over from the green wild species, or the red is the human one, which of course is the one that we're really more going to talk about today. But you can have a spillover from the green zoonotic virus pool into domestic animals and see amplification, and then a spillover into humans, which is what we think of when we think about what's happening with MERS, for example. So a spillover into camels, and then a cooking along in camels before we really detected and noticed that those camels were likely passing on their virus onto people. So what we want to do is we want to get in there early, and instead of sort of jumping in at the end of the red epidemic curve with some vaccine, and really often when we do that, frankly, I hate to say, it's too late, right? So it's a Band-Aid. We're throwing money at a problem, but it's almost a PR campaign, because unfortunately that thing that comes the next time is oh so rarely the thing that came the last time. So the vaccine is often too late. So what we want to do is get up in front of it. So go all the way back to the green, where we have these little viral, maybe little events, but often it doesn't make the host, the native host in which they've involved, the wild animals, sick. So we might not see disease in the wild animals, but we want to know what's out there and what might be the viruses that are in those animals that could spill over. Then what circumstances might they spill over, both to domestic animals and to people, so that if we know what's out there, and if we know how to detect them, which is a huge issue, then we would be able to detect more quickly and be prepared, maybe even with biological countermeasures, vaccines, and other things. So we want to actually be ready to jump in much faster with control, and then hopefully lower those curves, or if we're really optimistic, make them go away altogether. So what we did as a team, and again, this is a big consortium. I'll show you a slide with the different partners in a minute, but we started working on mathematical modeling and forecasting, and really what we saw, and I think many of you here who work in the field are aware, there isn't the data to do that forecasting. So if you ask the National Weather Service, how much data did you need before you could make a model to tell us within a reasonable certainty whether it's going to rain today? And did it match what they said? I don't know. Maybe not today, maybe tomorrow. But it took 50 years, right? 50 years of them collecting the baseline data in a great systematic way to be able to make the models that even started to get us to a place where we could forecast the weather. So we don't have that data. We have a bunch of science going on around the world, and maybe flu is our best example of where it's done the most systematically, but those of you that work in flu even know that that's not the case, right? It's often little different pockets of great researchers, and then depending on how they share their data, we get into some trouble, with patents and all sorts of things. So we did the modeling. What did the modeling tell us? It told us we didn't know a lot and that we needed more data. But we keep doing those efforts. As the data get better, our models get better. Those helped us to target key interfaces and species that will be the most likely locations and circumstances, and then the most likely hosts that people would interact with and have the opportunity to be exposed to a pathogen that could spill over. Then we had to get into the laboratory and actually detect them. Now, again, those of you working in the field know that if you don't have a pathogen to look for, there is never really a diagnostic test to run. Now we have metagenomics, but even metagenomics, we need the library, right? To be able to say, what are we looking for within this sample? So we don't have the technologies that we really need to be able to just detect anything that might be out there. We're getting there. We're getting there, but we're not quite there as far as the data on the backside that runs all the models. And we needed to identify both known viruses that can make people sick and unknown viruses that should go on that list that we should be thinking about. And then how do we share that information? Not only get that out to the public health community, like all of us here, but get that out to the governments that need to be thinking about how they run their programs and what they surveil in their countries. But just having that first conversation like I'm having with you, why is this veterinarian coming in and telling me, the Minister of Health, that I should be looking for these other crazy viruses when, oh, by the way, I don't have enough cash to really do a great job on TB diagnosis, right? That's a hard sell. Okay, and so what I thought would be that hard sell turned out to be kind of a different story, and I'd like to share that with you as we go. So this is the PREDICT project, the USAID-funded PREDICT project, and at Davis we lead a consortium, includes EcoHealth Alliance and Metabiota, Smithsonian Institution, Wildlife Conservation Society, as well as the Ministries of Health, Agriculture, and Environment in all of the 35 countries in which we've operationalized this project. Then we have some great laboratory and information management partners at Columbia, at Harvard and Boston Children's Hospital, HealthMap, ProMed, UC San Francisco. So the consortium is quite broad. And inside every country, we have implementing partners that may be the ministries or it may be a local university or NGO where we headquarter our operations. Now, what the modeling showed us that helped us to get started targeting is that we really needed to be looking at those species that we live closely with, humans live closely with, and that are closely related to us. So that would be the domestic animals and primates. We could have guessed that, probably. And then we also identified those taxa, especially rodents and bats, where we've seen being the source of the zoonotic agents that can spill over to us. So that was a good first guess of how we would target our efforts. And then we went out and we did it a little more broadly than that at the start, but we targeted then these species. Again, coming to talk with the ministers in the countries, you're going to look at monkeys and bats. I need you to look at, right, people. Okay, so we're lucky to also be able to partner with other folks like WHO and CDC and make a concerted agenda so that we are all balance each other out. And so we were really focused on this zoonotic pool. And in the end, folks were really happy because they see these emerging infectious disease events over and over, and having an opportunity to have partners come in so they could save all of their budget for the known diseases in their countries allowed us to say, let us work with you to fill this gap and help you identify with the science where you might want to add some focus, but not just coming in and saying, you're just doing, it too narrowly, you need to look at everything, because it's not practical. And so some of the work that we've been doing and continue to do keep reinforcing that bats, non-human primates, and rodents are really the place to be looking for these viruses, both historically from where we've seen epidemics and pandemics emerge, but also from our current data showing us the viral richness and the host jumping for those viruses. Now, this is another kind of modeling work that we've done, and this shows you the different interfaces. So not only can we map the different viruses that are shared among hosts, but we can map how viruses are shared among interfaces through which they might spill over. So by interfaces, I mean a rodent running around in your house, or a bat in a cave where you might be starting to mine. So those kinds of interfaces, if we see the same virus or viral family in those different interfaces that have spilled over into people, so this maps every red circle there is a virus that has spilled over into people from animals, and you can see that we get a lot of connectivity amongst the viruses when we're over on the side that's not vector-borne, so all those miscellaneous transmission routes and interfaces. So those are showing us that we need to pay some attention over here. The vector-borne, also super important. Tons of viruses that are transmitted vector-borne, but they tend to cluster with themselves. And so that was actually pretty good news for that balancing, because CDC has a rich history and great scientists working on the vector-borne side. So we were focused much more on this kind of messier side, where we see the sharing of viruses. And so that led us to new transmission interfaces. So not just what we could find in the literature, but all of these different new industries like bat guano farming. So if you make a bat a really nice condo, it will come and hang out in your yard if you live in Southeast Asia. So that's what these are, bat condos. And if you attract them, you can collect their guano, and guess what? You can put it on your crops directly, or you can sell it to others that are putting it on your crops. So you can see the folks down in the lower corner collecting that guano. Great PPE, right? So we know that many coronaviruses like SARS and MERS and things can be found in that guano. And but the people need to make a living. So can we help them figure out how to do things safely, or suggest that they might take another route of commerce than this one? So that's the kind of interfaces that we identified. Or alternatively, we test that and we tell them, you know what, you're right. This isn't so dangerous. There isn't a lot here. Or if you put it in UV, if you wear the protective equipment and get it out in the sun, you are going to kill almost everything that's in there, because then it is a viable and safe-ish occupation. So that's where we were going with some of this activity. So it's an iterative process. You figure out where you're going to target, you get out into the field, you get those samples into the laboratory safely so you can process them. In many places we didn't have laboratories to even work in because there weren't viral laboratories in the countries. Then you get that data back into the models so the models get better so you can continue to target. So while we were looking at those interfaces locally like bat guano, we also couldn't forget about the large-scale ecological drivers. So the biodiversity and changes in human population density and how we use land keep coming up as changes that we need to be watching. So when you disturb the ecology of a location, we see more likely interfaces coming up that can give you a chance for exposure. So I'm going to tell you now about our first 5 years of giving this a go. So we started with 20 countries, and in many of the countries where we started, the partners in the consortium did have some relationships already, had done some work in those countries. So we had a little head start to figure out how to talk to folks in those countries. But it was still a challenge. But we were able to get out and train people, again, our staff that we were hired from within the countries, in our partners and ministries. And in just 5 years, we were able to sample 56,000 animals in those 20 countries. And then we were able to also, or we had to, if you will, figure out how to get samples from species we couldn't capture and sample, either because they were revered, or they were endangered, or they were protected, or they were just too wily for us to be able to catch them. So this is a picture of a monkey where we've got a new technique that we validated in primate centers, and we're able to say what's the best kind of rope that we could throw, and what's the best kind of jam or peanut butter or attractant for the monkey to get him to chew on it and then release it so that we could pull it back and still get the same amount of virus or the same kinds of virus we would get if we just went in and swabbed that monkey. So we were able to create some of these kinds of sampling to get those samples, those critical samples, into the lab. Now, we had to develop a platform to get the diagnostics done, and that was super difficult because, again, we didn't know what we were looking for, right? And so we picked all the viral families that had ever caused an epidemic or a pandemic in the past, and we created a library of primers that could be used in a conventional PCR sense, so a very inexpensive platform that we could do in any country and in any laboratory by deactivating the sample so it wasn't dangerous to the technicians. We still use a lot of biosafety precautions, this biosafety cabinet, but we are able to move forward and test for the known and the unknown. So all the known viral diversity that's caused disease in people, but in those families find out what else is there. So we're not just testing for one Ebola virus, which is usually the way the tests work. We're testing for all the filoviruses that might be in the sample. And sometimes we had to build the lab. And this is where our scrappy teams, this is our Sokoine University of Agriculture team, really helped us out and said, okay, if we're going to have to build laboratories and you don't have any capital outlay funds, I know you're all familiar with federal funding, so we couldn't build anything. We could just outfit the laboratories. So they said, okay, we'll build the laboratories. This is our best example. They built a molecular virology laboratory out of 2 shipping containers with granite countertops and marble floors, and it cost them about $40,000 to put this whole molecular virology laboratory together to be able to do the work. It also separated our work, which could be with dangerous pathogens, even though we're deactivating them, from the rest of the university labs where they were teaching students and didn't have a lot of experienced people for their safety. And so these are the way that we put together the protocols and the viruses. We also had to make control material. So we couldn't ship around the most dangerous pathogens in the world to be the positive control material. So we had to synthesize control material. And the synthesized control material also allows us to say whether or not we get contamination in the lab, because when we pull up positives, we can detect whether or not it's the original virus or contamination of our control. So that helped us. So in the first 20 countries, we identified 32 laboratories that we thought we needed to work with to be able to get this work going. About 6 of those already could do the conventional PCR platform that we wanted, but the other 28 did not have experience with that. And at the end of 5 years, I think this is what I'm most proud of, is that 27 of those laboratories could detect and diagnose virus in their countries that they could not have done before. We ran over 400,000 diagnostic tests in those countries. So we try to do everything within the country with just support back here in the US with those labs that I mentioned before. And what did we find? Well, we detected over almost 1,000 viruses in the first 5 years. We're much over 1,000 viruses now. About 182 of those were known viruses, so bad actors that we were expecting, but maybe they didn't know they had in the country, or they didn't know that they had in that area at all. So we helped to identify where these viruses were occurring that they were worried about already. Also, 820 new viruses that are in those viral families that have the potential to spill over. So coronaviruses, flaviviruses, hantaviruses, all these different viral families. That helped us also target what we wanted to refine in the next 5 years. It also contributed to the science of ongoing problematic diseases, at least in our thought processes, like could SARS have directly spilled over from bats, or did it need to go through a civet, like in the market where SARS was found? We also found things like MERS-like viruses in those guano farms in Thailand and some of the other places. So we could also help to target some of the surveillance for MERS when MERS was first emerging. And then we did niche modeling that helped us look at, okay, given where we think MERS is, where the reservoirs likely are from the bats and where camel production is, how can we develop a hotspot map that tells us where we should be looking for the source of MERS? So this is the kind of data that improves our models and contributes to the global community, as well as allows us through the health diplomacy to go back and say, okay, and here's how you could safely do your surveillance if you're worried about MERS in your country. We also found diseases from humans spilling back or spilling over into endangered species populations. So this contributed a great deal to the conservation of certain endangered species, like the endangered mountain gorilla. Here we found actually we found mortality from 2 different human viruses while we were looking for those viruses that might spill over into people. And then we were able to start to get a handle on how you estimate the viral diversity that's out there. And that's really where we're going with the next phase, is that viral diversity that's out there, is it discoverable? Can we know more so that we can be informed? And just like I got in a car and drove here today, actually an Uber, and I did almost get in an accident. It was very dangerous, I have to tell you the truth. But that was more dangerous than me getting exposed to these viruses, right? But if I know that, if I know what my risk is from those viruses, I'm more likely to make informed decisions rather than running around and closing schools in Dallas because of Ebola, right? So that's where we need to go. We need to get into a different paradigm. And so we improve our global hotspot maps of where we think we should be looking for these viruses and all of those different factors, as I mentioned before, so that we can continue to refine our surveillance strategy and our targeted discovery strategy. In addition, we did outbreaks with the governments. So because we were working so closely with the governments and working on undiagnosed or previously undiagnosable diseases, they often asked us to help them out. And in the first 5 years, we supported 23 different outbreaks, again, with our colleagues from CDC, WHO, in some cases, and in other cases, they were so small, they were a small outbreak of just a few people, that we were able to help them get in the laboratory and process their samples before they knew if they needed to call in the international community because we're embedded there. And I think one of the cool stories that go along with the outbreak investigation is we saw places where they actually had rules and regulations in place where they couldn't release funds from the government to go and respond to an outbreak until they knew the cause. So most outbreaks were not being even investigated by their country with their country funds because they could never identify the cause until the international community came in, to a completely different paradigm where they have zoonotic disease task force that is set up in advance. It has members on it. It has environmental and animal protocols in it for the first 24 hours, so doing contact tracing, just like we trace contacts among humans, we also ask them those questions about how they're interacting with animals and the environment so we can identify the source. And then we get that information out to the public. So every sample, every positive, we clone and sequence and get as much information about the virus as we can. And we take that back to all 3 ministries, Ministry of Health, Agriculture, and Environment. And we get them to release the data to the public. Now, we set that as a standard. We said, we have to have your agreement that you will let us release this information to the public or we can't work here. Now, is that a legal agreement? Are we going to sue them if they suddenly say, don't release that information? No, our only stick is that we won't work there anymore. We won't help anymore. And so that has been perfectly lovely. We have not had a single country not let us release a bit of information. So once we have those viruses and they're released from the government, we get that information onto our health map site. And you can access this through the UC Davis just predict.global site or go into health map. It's a little harder to find that way. But you can go predict.UC Davis and you can click straight into health map. And you can get a surveillance layer that shows you everywhere where we've sampled, and you can zoom in and it'll tell you how many species and how many samples from each species. There's a test layer that'll tell you our test results. And that will give you the virus, how we've named it, what family it's in, what its most related virus is, if it's a new virus. And then for those that are already placed into GenBank, there's also a link that links you straight out to GenBank. So trying to get this information into the hands of the people who can use it. So this is kind of an example of that information flow. I think I've covered it for you already. But then what? So then we have a bunch of viruses. So we have 800 more than now of viruses. We have 100 new coronaviruses. Which ones are the next MERS? Which ones are the next SARS-like viruses? So how do we do that? So now we're working really hard to come up with that sort of risk ranking system. Will we be right? No, because no one's done it before. I was actually really angry because I thought, okay, I have these viruses now. Which ones? Global community. I'm pouring through all of the literature. How do we know which ones are bad? Nobody knows because nobody ever did this before, is what someone else said to me. Like, dummy. Why would anyone know if nobody ever went out and tried to find them all? So that's a big challenge, definitely. And so we're not just using the virologic information, we're also using that epidemiologic and ecologic information to try and come up with that risk ranking system. So stay tuned for that. And what about taking it to scale? Can we really start to look not just at the wildlife, but at the humans and at the domestic animals and see how much sharing there is? So that's what's really happened in this next phase of the what we call Predict 2, is that we know a lot more about what's out there in the bats, primates, and rodents, but not in every country in the world. We want to know that what are they sharing with the humans? So now we're testing in conjunction the humans at those interfaces to see how much sharing there really is, as well as working with FAO to look in the livestock. So that's the new global agenda, is look at all three in conjunction and see how much viral sharing and potentially start to forecast the viral jumping. That means we're looking at behavior, a lot of work looking at human behavior, not just the behavior of how do I pick up the guano and move it safely into the sun, but the behavior of where are we building roads, where are we investing in new industries that might take a whole toll on the ecological balance or turn things up and sort of stir up new viral spillover events. And so now many of you have seen, everybody knows this quote, right? Donald Rumsfeld. So it's a funny quote, we all laughed about it, but it really does help us to forecast where we're going next. There are unknowns. We know them. We know that they are unknown and we don't know, right? So what can we do? What can we do? Can we take this 20-country effort with just a few species and locations and take it to scale? Well, we know that the human population is rapidly increasing. We have people pushing out into wilder areas and bringing with them their domestic animals, which, as I mentioned, could be a mixing pot and cause productivity problems in the domestic animals, economic losses, but also changes and spillover into people. So all of these things, crop production, resource extraction, start to change the environment and push us out into those areas. So we have increasing animal-human contact. And then potentially, likely, what we've seen historically over the last 20 years is increasing contact and spillover rate. And so what we really think that we can do, and this is the brainchild of Dennis Carroll at USAID, is to take this take this whole baby to scale. And instead of just looking in the few areas that we believe by our models to be the most dangerous, really start to look across the board, across the world. So if we take here that we have these hosts, we have all the possible hosts in the world. So the species richness, if you will, they're just the ones we've identified so far, but the species richness of viral hosts there depicted in the globe, you can see that we're really concerned about the vertebrates. Those are the ones that seem to spill over into us, right? So if we're concerned about the vertebrates, we've already taken out a whole bunch of noise and said, we're not going to focus there. We're going to focus on the vertebrates. And if we focus on the vertebrates, we can even narrow that down while continuing to analyze and model, as I mentioned. We know non-human primates. We also know birds for influenza. We know bats for the coronaviruses. So we can start to narrow the taxa. So we don't need to look at all the vertebrates, right? We can start to narrow our task. Again, getting to something that's practical. And then we get to the viral diversity. Well, what about all of the viruses that might be in those hosts? Can we narrow that down? Well, we think we can. There's somewhere between 16 and 22 viral families that have the ability to jump into people so far as we know. Now, we're continuing to look. We're not going to just stop looking altogether for any new ones, but we're doing a pretty good job diagnosing human disease now in at least places like this, right? And so if we can do that, that helps to give us that information in this global information age that helps us narrow the viruses. So if we can get the hosts down to a narrow amount, the viruses down to a narrow amount, then even from those viral families, only a small proportion are really going to spill over into people. We think we can detect that. We think it's on the order of less than $10 billion, maybe only about $3 billion that it would cost to detect all that viral diversity that can spill over into people. And oh, by the way, every time we have a huge outbreak like the Ebola and the Zika, we spend on that order. Okay, so the World Bank estimates that we could save $6.8 billion a year by preventing zoonotic diseases. Okay, so we're talking about the amount that we spend in a year that we could be saved in 1 year to discover all the rest of the things that we should be worrying about that's out there. And so that's where we're hoping to build up some momentum and get some enthusiasm from the global community to participate in identifying these unknown high consequence viruses so that we stop chasing things when it's too late and start preparing for those most likely worrisome things and maybe have an end to the pandemic era. So that's really what I wanted to talk to you about today and leave you with that hopefully a little bit of interest, if not excitement, for a proactive paradigm. And I'm happy to take any questions. Come with me.
So we know that, or at least I think we know, that we're often, what we don't know, but we're often concerned about the fact of what's happening, for example, with the flu viruses. Mm-hmm. That is that they have seem to have a huge amount of activity of combining and changing and whatever, and then one day they could change into the one that is transmitted more rapidly or becomes much more virulent than the ones before for whatever reasons. I've already said more than I know about this. But it seems to me that that's an issue, and what about this issue about the repetitive monitoring of, the bird market in Thailand or whatever? Right. The issue of trying to pick up on what's really happening with these things and more proactively out in the animal population that has normally been a vector transmitting to people, trying to pick up on what's happening with these things.
Yeah, so some of these are smarter than us, right? So we need to change the way we do things, in my opinion. So it's a great point. Flu is just a nasty bug that wants to keep recombining and changing, so we chase it. We chase it and we chase it and we try. The best minds in the world in this field are working on flu, and they're trying to figure out what should be in the next vaccine, and they're trying to figure out how to do this. I think that's great. We can't stop it, right? It's what kills a lot of us every single year. But wouldn't it be great if we got in front of that and instead know what the viruses are that are out there, what flu viruses could recombine. People are working on that. It's very expensive, right, to go flu virus by flu virus. And so what about figuring out how to prevent the exposure, right? And that's the work that you're indicating, like, do we rotate markets and do we close markets and how do we clean? But I think, one story that I can tell you is during the H7N9 outbreak in China. We were working in China and we were working with the Guangdong CDC and others. And we were able to go in, and while they were using a real-time test developed rapidly for the H7N9, we ran our protocols and were able to show co-infected in the same individuals other H9s and other things that had the potential to be more human-to-human transmitted. So if we know that, if we're doing that work real time, as opposed to just now we're so busy responding to this, if we knew all the different viruses that were in that market, then we would be more likely to be able to say, hey, this market is a dangerous one. Let's not do this market. Let's figure out what's happening in these markets where there's not all those different viruses, flu viruses. Yeah.
Hi. In your presentation, you mentioned that you alluded to past pandemics and past epidemics. How do you and your team learn about those past events? What media do you turn to learn about those past events, aside from the obvious databases? And how do you incorporate those historical lessons learned or not learned in your contemporary research, your practice?
So we've really worked on the time frame that has the best data. So the last 40 years. The historical information before that is too sketchy to be able to really build models from. So we really use the last 40 years and even more carefully the last 20 years. Then actually we're not using, we of course use GenBank and the flu networks, but we also go straight back to the primary literature and if they're alive, the investigator, because we've been disappointed about what ends up in those databases. Now, the databases are great, but we are still not across the board sharing what we all think should go into those, right? I'm preaching to the choir on this one. But from GenBank specifically, I think we calculated for the zoonotic viruses, it was less than 1% had a geolocation. So it's very hard for me to make a map if no one will put the location in. And we've had to deal with our countries and say, okay, we want to say we found this at this location, we want to put this in this map. We had to deal with how many decimal points on the longitude and latitude are we going to go to? Because do we want to say it's at this person's stall in this market? Or do we want to say it's in this tourist town? Or do we want to be a little more broad, and say it's this region because we don't want to impact artificially or inappropriately economics? And so that's been part of the conversation. But really it's going back all the way to the primary literature and to the investigator themselves to say, did you really document a spillover? Did you just find a virus that in a person and in a dog, or did you document that that person knew that dog, right, and ever would have contacted? So we're trying to get back to the primary information, unfortunately. But working with FAO specifically, I hope WHO will come into it. We're trying to come up with those fields that we think we all agree we want to know about so that we can improve the databases in the long term.
Well, congratulations on your work. It's really wonderful and important and is very well presented by you today.
Thank you.
Thanks. My respect too. You didn't comment on the futility of trying to grow flu vaccine in eggs. And that just is nonsense. Who's working? Where do we stand on getting?
You want to get me in trouble? No, that's not my field. So, so I am gonna bob and weave. It's not my field, but I believe, if I can sort of take it to a more general level. I believe that if we can start to move our diagnostic platforms to something more broad, so instead of virus by virus, we go more broadly to detecting all the diversity in a viral family, that we should be able to, now people have been trying, I'm not saying this is an easy step, we should be able to also get to vaccines and treatments that are better, that are more broad than virus by virus, or even have them at all, frankly, because we don't really have good treatments, very many for viruses at all. So prevention and treatment, I believe, if we're smarter than we've been, could get us to that more general place. Because I think we've gone so far down the rabbit hole of targeting tests and treatments to very specific, we love genetics because we learn so much about it, and it is great, and it is helping us, but our desire to go so specific, sometimes we lose the forest for the trees. It's the general practitioner versus the specialist, right? But if we all have 20 specialists working on us and nobody's looking to say like that we're just can't take another drug because we lose the general practitioner, I feel like we have we're risking that in this realm as well, and we need to step back.
I mean, it's a major problem, whether it's CDC or WHO or anyone else, by the time they blow the whistle, the virus is not stopped mutating, and it takes, N months to produce the vaccine in volume. I mean, it's a
Correct.
crazy system.
I actually took the flu slides out where I showed how long it took for the H1N1 flu vaccine to basically more than a quarter of the world was infected before we had the vaccine. So you are correct, sir. What we do about it, I think, remains to be the subject. That's my goal, is just to get these dialogues started so that we can move the line. And I think USAID, I would really say, is being very holistic, using the One Health animal, human, environment perspective to say we have to do this differently because, oh, by the way, they're the ones that provide that vaccine often around the world. They pay for it outside of this country in the developing world, and we deliver it as part of our health diplomacy. That's very expensive, and if it's not necessarily working because it's too late, then there are a lot of people that want to change that and not just throw that money away.
Are there people as smart as you?
There are a lot of people smarter than me. Yeah. There's people that are interested, but I think this is not the way we're trained, right? We're not trained in our graduate programs and things. We're trained to take little bits and pieces off, which is really important. We need that basic information to be able to step back. I think we need to train at least open the minds, and I think our next generation is opening their minds to say, yes, I want to be really good at this tiny little enzyme piece, but I also need to think about where it fits in the big picture. And frankly, the funders are pushing us in that way because they didn't used to ask us, what are you going to do with this information? And now they do, which is a good thing.
And they may be paying more attention to the fact that it actually does get deposited and shared as well.
Yes, absolutely.
Which is good.
Yes, sir.
Can you say a little bit about your geographic coverage and how you ended up in the countries that you're engaging with now and which you haven't been able to that you've tried, that kind of thing, please?
Sure. So I think it was a combination of science and politics for the first 20 countries. So a combination of the first hotspot map, which I said, is the best we could do, but needs to be better. Telling us that the tropics, where wildlife diversity and human population density were greatest, that's really what drove that. And then saying, okay, but there's a lot of those, and USAID saying this is where we'd like to make our efforts. This is where we're willing to make investments because of safety and relationships with the U.S. government and all of that. That changed dramatically about a year and a half ago with the Global Health Security Agenda. And so our countries now are quite driven by the Global Health Security Agenda Phase 1 and Phase 2 countries. They happen to be the same kind of demographics, though. So they're developing countries, mostly in the tropics, a little bit elsewhere, where those same factors are happening. And then we're adding in constantly improving. So as an example, West Africa and the actual spot where we believe this most recent West African Ebola outbreak spillover happened, that was bright red on our map. And around it was less red, but we weren't working in West Africa in the first phase, not because we didn't predict that it was important, just because you had to start somewhere. That's where USAID wanted to go. Josh?
Yeah, I know you guys are primarily focused, I'm a great fan of this work, by the way, I think it's fantastic. And I know you're primarily focused on the emergent viruses and novel discovery efforts. What portion of your efforts do you think going forward are going to be dedicated to thinking about risk assessment for known pathogens? I mean, Ebola and Zika were both known for many years,
Right.
Zika not so prominently, but
Right.
those are the really disruptive ones. SARS perhaps is an exception to the rule.
Right. Yeah, so I would say we're non-discriminating, so we don't really care if they're known or not known. Our bread is buttered in the emerging piece, but when I think about emerging, it's not emerging meaning never discovered before. They're all out there. We're just happy, we're so sort of arrogant to think we discovered it. Well, the virus probably doesn't think it got discovered, right? I think it's more the mapping of the different viruses that we think are riskiest. So figuring out what makes them risky. So things like Ebola and Zika help us to learn about how we really rank them as far as their risk. And again, we're going to rank many as risky that aren't risky. And that's going to put, because we have to be conservative, right? That's going to put us in jeopardy, because a lot of people will throw stones and say, oh, you said all these 100 were risky and really only one of those has caused disease since you started or whatever. That's you just have to be willing to take that hit and just not, have a thick skin, not worry about it. But I want to know where all the Ebolas are. I want to know what the gosh darn host of Ebola is, right? We still don't know that. We can make a really good educated guess, but we haven't been able to document it. So I want to know where it is and what different versions of it are out there and in what hosts. And so then you could say, hey, best not to build a restaurant under this tree, right? Really, that's the practical application. And I'm a conservationist at heart. I'm a vet. I trained in conservation, and I do believe that biodiversity is critical to us and our survival, but we also alter our landscapes quite a bit and manage it, and so we can manage our risk, I think. Yeah.
What are the circumstances that lead a non-pathogenic virus to become pathogenic, number one? And number two, are all the viruses that ever existed present now, or are new viruses formed?
Oh, most certainly new viruses are being formed. It depends how we call them. So that's been a tricky piece of this, is how do we decide what's a new species versus a strain of an old species? Is it a new virus? Is it a known virus? It just has some mutations, substitutions, deletions. So I wouldn't be arrogant enough to say that, well, I know they're not, they just can't all be here, the ones that are going to be here, because we see new things happening all the time. Whether we call them new species or not becomes a big question. What causes them to become pathogenic? Boy, if we could answer that. That's what this is about, is getting to that. So if we know all of the ones that are out there and we know which ones are pathogenic so far, and we can find the traits that are consistent across them, that will help us decide that, help us push us in the direction of knowing that. Right now, we really, really don't know. There's some substitutions in Zika that happened in French Polynesia that indicate that it did change and that it probably did become a little more pathogenic on its way across the earth. But again, which ones of those is it virus specific? Is it a combination of what's happening in the host? That's why we're trying to not just look at it virologically, but also look at it from an epidemiologic and ecologic perspective. Yeah.
I just had a quick question, because you had briefly mentioned about kind of the interdisciplinary nature of One Health, and I was curious if you could talk a little bit more about the training efforts, especially for like young veterinarians and young medical students.
Sure.
And do they interact? And
Sure. So I can tell you it's easier in developing countries because they're less siloed than we are. And they deal with the problems that are the easier examples to talk about with One Health, like emerging and re-emerging and expanding diseases on a more personal and practical and daily basis. We certainly partner. USAID has also invested in that in the One Health workforce, and so University of Minnesota and Tufts lead that consortium, similar to the PREDICT consortium, to help with universities, again, just in those target countries, not here. And there also is another piece called Preparedness and Response that's working in governments to help expand the One Health kind of perspective. So that's in those pieces. Here in the US and in Europe, I think the students are demanding it, and that's a great thing. So we're really seeing a push from the students. We have a very active Students for One Health club, and we have a symposium on One Health, and we have law students, medical students, and vet students, as well as some ecology, and now we're getting the social sciences. We're trying really hard to get them involved. But is it driven by the faculty? Less than by the students, though you are seeing some expanding programs. So we have our One Health Institute. We're seeing those pop up in other places like Penn, Colorado, Texas A&M. So you're starting to see those things. So it's coming. There's no chance it won't.
And we need to get the informatics and information professions into that One Health thing as well.
Yeah.
We
do. We have a whole, I didn't mean to, we have a huge informatics team. So
I just wanted to say that the group that John Brownstein, the group that originally developed Health Map, that was with a grant from the National Library of Medicine. So we're very proud of that. And we're going to step over here so we can get our picture taken. Well, I give you this wonderful plaque, I'm sure, or certificate as the 2016 Leiter Lecture, and a great one. Thank you very much.
Thank you.
As Glenn mentioned, the Friends of the NLM are treating us to a nice reception outside, so I hope we'll see everyone there. And we're sorry that all of you who are watching remotely can't come out and have a cookie with us, but we'll have a second one for you.